Module 20, Chapter 25, your digestive system. Now, as you know, I'm trying to keep the online course equal in rigor and expectation as an on-campus course. And in an on-campus course, the functions of the liver are independent study. And in keeping things even, the functions of the liver are also independent study for the online course. Now these functions are very nicely summarized for you in Table 26.6 on page 1016. Your essay question on exam 4 is going to be to list and describe a certain number of functions of the liver. Now there is a Get to Know Your Liver homework assignment in Canvas that is also going to help you work through this content as well. And please let me know if you have any questions as you are completing the independent study. Now, your digestive system consists of two groups of organs. You have what is called the digestive tract. The digestive tract is also referred to as the gastrointestinal tract and the alimentary canal. And the structures included in the digestive tract are the mouth, your esophagus, your stomach, your small intestine, and your large intestine. Working with the digestive tract, though not part of it, are various accessory structures. Your accessory structures will include things like your teeth, your tongue, your salivary glands, your liver, your pancreas, and your gallbladder. Now, the digestive system is going to actually perform a variety of different functions, most of which involve the acquisition of nutrients for your body. So, for example, your digestive system is involved in ingestion. Ingestion involves the intake of food, and this is selective and does involve conscious choice. So you are making the decision to ingest that food. It's then going to involve digestion, which is the breakdown of the food that you ingested into usable forms. Now there are two types of digestion that will occur. Mechanical digestion is a physical breakdown of the food. That means you're going to take a giant bite of a hamburger and you're going to use your teeth to basically rip that large chunk of burger into smaller components. So it involves a lot of cutting, grinding, churning. You're not breaking down the chemical structure of your food. You're just breaking down a big food chunk into smaller food chunks. The breakdown chemically of your food is called chemical digestion. This is when you're going to use digestive enzymes to help take your big macromolecules, which are things like proteins, triglycerides, starch, glycogen, nucleic acids, and break those really big macromolecules down into their building blocks. Now, at this point, I'd like to remind you that if you're having any kind of memory lapse with regards to the macromolecules and what their building blocks are, you need to go back into Chapter 2 from A&P 1 and you need to start reviewing that information. Now, once you have mechanically and chemically broken down your food into usable nutrients, the next thing that your digestive tract is going to do is it's going to absorb those nutrients. Absorption is the uptake of nutrients into the epithelial cells lining your digestive tract, and then moving those nutrients from the epithelium into your blood and lymph. After everything that is usable has been absorbed, you're going to undergo compaction, which is where you're going to start absorbing water and consolidating the remaining residue in your digestive tract into feces. once you have your fecal matter formed, the next function is defecation, which is the elimination of those feces into your environment. Now, those are sort of the hallmark features of the digestive system that most people are aware of. However, your digestive system is also very protective. It has the ability to protect itself from the corrosive effects of all the enzymes and acids within its lumen. It has the ability to protect itself from mechanical stress and abrasion. And it has the ability to protect you from the bacteria that are either ingested with your food or are residing within the digestive tract itself. The activities of your digestive tract are regulated. Some of the mechanisms are neural. The neural mechanisms are going to include your short or myenteric reflexes and your long or vagovagal reflexes. Now, the short reflexes are going to utilize your enteric nervous system. In A&P1, when you were introduced to the nervous system, the enteric nervous system was mentioned in terms of it being a neuronal network associated with the wall of your digestive tract. The actual nerve plexus is called the myenteric plexus. So if you have any stretching or chemical stimulation of the tract, this plexus is going to be activated, and when it's activated, it's going to stimulate contraction within the wall of the digestive tract. Your long reflexes are going to act through the autonomic nervous system. You're going to have nerve fibers that are going to carry sensory information from your GI tract to your brainstem. Your brainstem is then going to analyze that incoming sensory information. It's going to create a motor command that it's then going to send back to the tract to create a response. Now, it's the parasympathetic division and its signals that are especially important in stimulating both motility and secretion of the digestive tract. There are some hormonal mechanisms that we will be mentioning as we go through the next few modules. These hormones are actually produced by the GI tract itself, and then they're carried by your blood to other parts of the digestive tract. For example, you're going to learn about the hormones gastrin and secretin. You also have some paracrine mechanisms. The main chemicals used for the paracrine signaling are histamine and prostaglandin. And remember, paracrine mechanisms are going to affect cells that are near the actual site of chemical production. So these are those local hormones affecting local function. Now, when you go to move material through the digestive tract, there are two major movements that are going to occur. One is called peristalsis, and one is called segmentation. Now, both of these are going to involve the visceral smooth muscle, forming the muscular layers of your digestive tract. Now, I want to talk about peristalsis first, which is what is shown on the right-hand side of your screen. In peristalsis, you are going to have waves of muscular contraction. These waves begin at the esophagus and proceed through the esophagus, through the stomach, through the small intestine, and through the large intestine as a giant wave. And the job of this wave of muscular contraction is to propel the contents within the digestive tract towards the anus. So this wave is maintaining one-way movement through the digestive tract. So again, you're going to have this wave in your esophagus as you swallow a oval mass called a bolus. It helps to move a soupy acidic mixture called chyme through the stomach and your small intestine, and then what is called residue throughout your large intestine. Now, even though this is normally in one direction, when you vomit, things are going to move in a different direction. But for the most part, in a healthy individual, one direction only. If you would like to simulate peristalsis at home, it is very similar to squeezing toothpaste out of a toothpaste tube. So imagine you open up a brand new tube of toothpaste and you have the opening where the toothpaste comes out at one end, and then you have the closed end on the other. So I want you to imagine applying pressure to the closed end of the toothpaste tube. And when you squeeze the end of the toothpaste tube, you push all the toothpaste in the tube towards the opening. And then as you use the toothpaste every day, you are repetitively squeezing from the end of the tube towards the opening of the tube, and you're constantly pushing the toothpaste ahead of your fingers out the opening. And that's sort of what is happening in your digestive system. You're using these little contractions to push the material within your tract towards the opening. Now, segmentation is a little bit different. Segmentation is not used for propulsion. So it's not used to help propel your contents towards the anus. However, what segmentation is going to allow you to do is it's going to provide you with localized cycles of contraction. So what that means is you'll be moving your contents through peristalsis along the tube, and then every few moments, peristalsis will pause. And in the area where propulsion has stopped, you will have localized muscle contraction occur to help churn and fragment your bolus and mix the contents of your tract together. So you're going to have peristalsis moving you bit by bit along the tract, every once in a while to pause to churn and slosh around the contents to mix them up for better digestion as you're moving along. All right, so how we're going to approach this particular system is we're going to start at the beginning, start where we ingest our food, and then just work our way very slowly to the end. So we're going to start with your mouth. Your mouth is also referred to as the oral cavity or the buccal cavity. Now, your mouth is the site of what is called mastication. Mastication is the biological term for chewing. Now, chewing is a type of mechanical digestion. You're not chemically breaking down macromolecules, you're just kind of ripping your food into smaller and smaller pieces. Chewing involves your teeth, but it also does involve your tongue because your tongue is responsible for moving food back and forth between your teeth. Now, the result of mastication is the transformation of your food into something called a bolus. The bolus is a very soft, flexible, sort of egg-shaped mass, which is going to be a lot easier to swallow than a large, irregular, firmer piece of food. Now, associated with your mouth are glands called salivary glands. Their role is to produce what is called saliva. Now, on an average day, a healthy person produces between 1 to 1.5 liters of saliva. There are two classes of salivary glands. You have what are called intrinsic salivary glands. So if you think intrinsic implies internal, these are salivary glands that are actually within the oral cavity itself. So they're in there with all of your other oral tissues. The ones that we're going to be kind of focusing in on are glands called lingual glands. The lingual glands are actually associated with the mucosa covering your tongue. There are also labial glands, which line the inside of the lips. However, we don't include those as part of our curriculum. You then have what are called extrinsic salivary glands. These are glands that reside outside of the oral cavity, but still deliver their secretion into the oral cavity. Now, there are 3 pairs of these extrinsic salivary glands. You'll be naming and identifying all 3 pairs for your digestive lab. You have the parotid gland, which lies just anterior to your ear on the external surface of your masseter. You have the submandibular gland, which is positioned just inferior to the mandible, and the sublingual gland, which is just inferior to the tongue. Now, the saliva that you produce is between 97 to 99.5% water. The rest of the volume is made out of the solutes, which have a variety of different functions. Now, the overall pH of your saliva is between 6.8 to 7, so it's almost neutral. Okay, very, very close. With regards to the different components, you have mucus. Mucus, as you know from A&P1, is a great lubricant. So it lubricates the entire oral cavity, which protects the oral cavity from abrasive forces while you chew. It also is going to bind your food particles together to help form your bolus and lubricate the bolus so it's easier to swallow. You then have various electrolytes, so your ions. So you have things like sodium, potassium, chloride. You also have phosphate ions and bicarbonate ions. Now, the bicarbonate ions are really important to note because bicarbonate ions are alkaline. And bicarbonate ions have the ability to buffer acidic foods. So a lot of the food that you eat is very acidic and potentially damaging to the structures of the mouth and the esophagus. So the bicarbonate can help neutralize those acids. You also can use the bicarbonate to prevent the buildup of acids that are produced by bacterial action. So you have a lot of bacteria residing in the mouth. These bacteria feed primarily off of the sugars in your food. Now, as a thank you gift for the food that you give them when you eat, they put waste on the surfaces of the oral cavity, especially your teeth, where you can trap food particles and you have more of the bacteria. And the waste that they deposit is acidic. So it's the bacterial acids that can erode your tooth enamel and cause cavities. And the bicarb in your saliva helps to neutralize some of those acids from your bacteria. Now, the action of all of this buffering is that your mouth stays pretty much right around 7.0, so it stays fairly neutral. You then have what is called immunoglobulin A. Immunoglobulin A is actually listed in table 21.3 on page 834. The function of immunoglobulin A is to help prevent microbial attachment. So it's an antibody, and it's going to keep microbes in the food you ingest from attaching to the lining of the oral cavity, which is going to help prevent infection. It also means that it's going to help you swallow those microbes to the stomach, where stomach acid most likely can destroy them. You have your first digestive enzyme that we're going to talk about in here also. It's called salivary amylase. Now, salivary amylase is going to help you break down your polysaccharides. Remember, your major polysaccharides in your diet are starch and glycogen. And thanks to the actions of salivary amylase, you're going to chop up those big polysaccharides into maltose, which is a disaccharide, and some slightly larger oligosaccharides. So salivary amylase is going to begin the breakdown of those big carbohydrates, but it doesn't finish it. Maltose and oligosaccharides are still too big to absorb. Now, salivary amylase is going to be swallowed along with the bolus, as are all of the other secretions, except salivary amylase is going to be functional between pH 6.5 and pH 7.5. So when you swallow salivary amylase and it hits that acidic environment within the stomach, it's going to get deactivated very quickly. In fact, once your pH gets below 4.5, salivary amylase completely becomes non-functional. You have lysozyme in your saliva. Remember, lysozyme is a chemical that can actually kill certain types of bacteria. Doesn't kill every type of bacteria, but it kills a good number. You also have another enzyme, it's called lingual lipase. It's going to be produced by those lingual glands on the tongue. They were a type of intrinsic salivary gland. Lingual lipase is going to help you break down your triglycerides into fatty acids and monoglycerides. However, lingual lipase is a very minor lipid digesting enzyme, so it breaks down a little bit, but it's not your primary lipid digester. Now, lingual lipase is going to be active between pH 3 and pH 6. So lingual lipase is made by the lingual glands on the tongue, but it usually does not function in the oral cavity because the oral cavity is kept pretty neutral, and this enzyme is active at an acidic pH. So it's going to be made by the tongue, it's going to be put into the bolus. Once lingual lipase enters the stomach, that acidic environment is then going to activate the enzyme so that it can do its job. Now, your saliva does have a generic flushing function where it is mostly water, so you can swish your saliva around in your mouth and you can use it to flush food particles off your teeth, off the inside surfaces of your cheeks, so general cleansing. You need saliva for your sense of taste. In order for taste molecules to interact with your taste buds on your tongue, they have to be dissolved in water. So if you want to experiment with this, go home. If you're listening to this at school or at work, if you are home, stick your tongue out, get your tongue nice and dry, and dump some sugar or salt on it. And I can pretty much guarantee you that if your tongue is dry, you will not be able to taste anything that you put on it. But the moment you put your tongue in your mouth and you let whatever you've placed on your tongue mix with some saliva, let it dissolve in some water, your taste will be right there. You also can use your saliva to remove waste molecules. You can put into your saliva things like urea and uric acid. They're the exact same waste products that are in your urine. Now, this is the long route to get rid of it, but if you put those wastes in with your bolus, you're going to swallow the bolus. Those wastes will pass through your entire GI tract and eventually be removed from the body with your feces. So, in essence, you pee into your mouth. In A&P1, I say this when we do sweat glands as well, because your sweat glands are also a solution containing things like urea and uric acid, and you can use it for waste removal. Also within the oral cavity, you do have your tongue. Now your tongue is composed of skeletal muscle, meaning if you want to move your tongue, you can make it do whatever you want. So you do have the ability to control it, although most of the time the control of its muscular contraction is subconscious. It has a lot of different functions. For example, it's going to be very important in speech because it helps to shape the movement of air through the oral cavity to help you make the different sounds that you need to create words. One year, a long time ago, I had a student in A&P2 who actually had a degree in speech therapy. And he brought it up in lab. And I, of course, don't have a degree in speech therapy, so it was wonderful to have someone explaining all the different mechanics of sound production. And I remember him saying that there are like 14 different movements that have to occur within the mouth to make the sound for the letter S. So there's a lot of very intricately controlled movement happening with your tongue and your cheeks and lips. Your tongue is also your primary location for your taste buds. You have small elevations on the surface of the tongue called lingual papillae, and it's on these papillae where you do have those taste receptors, those taste buds. There are some taste buds on your epiglottis, which is the little flap that is going to help to cover your airway when you swallow, but primarily taste buds are on, the majority are on your tongue. Your tongue also is involved with detecting touch and temperature, which is important for when you are eating. You actually use the sense of touch to help gauge how strong your bite force needs to be when chewing your food. You use your tongue to help manipulate food when you're chewing, but also when you're swallowing. It's going to help guide the bolus towards your pharynx. And again, it is where you do have some of those intrinsic salivary glands, like the lingual glands, that make your lingual lipase. Now, once you have your bolus and you go to swallow, that bolus is going to enter your pharynx. Now, the pharynx, you learned in the respiratory lab, is going to help to carry air from the nasal cavity down into your larynx. Well, the pharynx actually has a dual role. It plays a role in the respiratory and the digestive systems. This muscular passageway extends from the posterior portion of the nasal cavity, posterior to the oral cavity, posterior to your larynx, and it ends where it connects to your esophagus. And it is divided into three sections. You have the nasopharynx, which is posterior to the nasal cavity. Usually that particular section is only involved in the respiratory system. You then have the oropharynx, which is posterior to the oral cavity, and the laryngopharynx, which is posterior to the larynx. Now, in addition to passing air, your pharynx does play a role in deglutition, which is swallowing. So it's going to help to guide your bolus down into your esophagus. Now, your esophagus doesn't produce any major secretions other than some mucus. And this mucus is going to help lubricate your esophagus, which is going to make it easier for you to move a bolus through that tube down to your stomach. The mucus also protects the lining of your esophagus from abrasion as the bolus is being transported. Now, in addition to that information, you do need to know that there is a disease associated with the esophagus. It's called gastroesophageal reflux disease, which is abbreviated GERD. Now, GERD is a reflux of stomach contents through what is called the lower esophageal sphincter into your esophagus. The lower esophageal sphincter, which is abbreviated LES, is a constrictor at the superior end or the entrance into your stomach. And this LES is going to help to keep the acidic contents of the stomach in the stomach and prevent them from refluxing up into your esophagus. However, in GERD, this does not work, and you are going to have stomach contents moving up into your esophagus. Now, your esophagus does not have the acid protection that your stomach does. So when this acid from your stomach goes into the esophagus, it actually starts to burn the lining of your esophagus, and it creates this really sharp burning sensation that is seemingly just posterior to the heart. So a lot of people will immediately think they're having some kind of a heart issue. But the esophagus is posterior to the heart, which is why that pain is so central in the thoracic cavity. And because of the burning sensation, people usually call it heartburn. Now, this leakage is because the LES doesn't close adequately. And there are a lot of different reasons for why that might happen. For example, alcohol and smoking both relax the lower esophageal sphincter, so it causes it to open up, allowing more easy reflux of contents. If you happen to have excessive acid production in the stomach, which could be caused due to acidic foods, when they get into the stomach, increase your acid production. Acid production can be elevated though due to stress. It can actually force this excess acid can force the sphincter to open. The valve itself could just be faulty. So there just could be issues with the lower esophageal sphincter. Now, they do recommend if you have GERD to avoid foods and beverages that could increase your acid production. So they'll tell you to avoid things like coffee and chocolate, tomatoes, fatty foods, orange juice, peppermint and spearmint, onions, all of these foods of which, are one or more are part of my daily diet. I don't know about you, all increase acid production. So by cutting those foods out, you'll lower your acid production, which is going to help avoid symptoms of heartburn. But there are a lot of different medications that can help as well. I'm sure you've all seen um, commercials for things like Prilosec. Those medications usually suppress acid production in the stomach, so it chemically decreases acid production, which is why they're so effective at alleviating the symptoms of heartburn. Now, before we go forward, I do want to mention that lower esophageal sphincter, and I most likely will mention this in lab, but just in case I do forget to mention it, I wanted to be sure I had it here. The lower esophageal sphincter is a physiological sphincter, and what that means is if you dissect a cadaver, there is no muscular ring around the entrance to the stomach. So if you go to that superior opening in a cadaver, there is no muscular sphincter. So they're not really sure what causes it. There is some suggestion that the smooth muscle in the wall of the esophagus in this area has a higher degree of muscle tone, and that's what causes the sphincter. There's also some evidence showing that the esophagus, as it goes through the diaphragm into the abdominal cavity, the muscle of the diaphragm surrounding the esophagus might have a different level of muscle tone, and it might be the diaphragm that's actually compressing the esophagus and forming the sphincter. Again, they're really not sure what the actual cause is, but it's physiological. It's not anatomical. So, since we have now swallowed our bolus, we've gone through the pharynx, through the esophagus, your bolus now passes through the lower esophageal sphincter into your stomach. Now, the stomach is a muscular organ. In fact, it is unique from all of the structures of your digestive tract. So, from the esophagus, from the small intestine, from the large intestine, in that your stomach has 3 layers of smooth muscle in its wall. The other portions of the digestive tract only have 2. So this is your most muscular GI tract organ. And it's going to have a variety of different functions. One is just bulk storage of ingested food. You eat a lot at one sitting. You eat, even if it's just a small snack, you eat a volume that if it was to move at that volume into the small intestine, your small intestine would actually rupture. So your stomach, with its ability to expand, can actually hold a very large volume of food from your meal and very slowly pass that ingested food into the small intestine at a small enough volume that you don't damage your small intestine. It's estimated that you only move 3 milliliters of food from the stomach and into the small intestine at a time. And if you think about how much you eat at once, it's a whole lot more than 3 milliliters. Now, because it is so muscular, your stomach is actually capable of a lot of grinding and churning of the contents that are in there. So there's a lot of mechanical digestion that occurs. There's also a good bit of chemical digestion thanks to the acid in the stomach and enzymes that are in there. In particular, your stomach is where you begin your protein digestion. And we're going to be talking a little bit about that in a few moments. Your stomach produces a chemical called intrinsic factor, which is going to be necessary for you to absorb vitamin B12. And its actions are going to take that oval-shaped bolus and transform it into something called chyme. Chyme is a highly acidic, soupy mixture of partially digested food. And it's this soupy mixture that's going to be able to flow more easily through your small and large intestines. Now, as much as it sickens me to say it, you have probably all seen chyme at some point, because everyone at some point has vomited. Now, if you think about the last time you threw up, and everyone looks, you throw up, it's natural, you look to see what's in the bowl, and you notice how it was always mostly water, but then there'd be the little chunky bits in there, and then the more you vomit, it might get a little bit thicker with some more chunky bits. What you're throwing up is chyme. So that's sort of what your stomach is going to turn your bite of hamburger into. It's going to turn into nasty, soupy, partially digested mix. Now, within the lining of your stomach, you have what are called gastric glands. Now, your gastric glands are going to produce between 2 to 3 liters of gastric juice per day. And again, this juice, like your saliva, is mostly water. However, it is going to have a lot of really interesting solutes. Now, if you think about the glands, your glands are going to contain several different types of cells. You've got regenerative cells, which are stem cells. These regenerative cells are going to provide a continuous supply of new cells. I mean, if you think about it, the lining of your stomach is highly acidic, So you're going to need to be able to regenerate that lining as it becomes damaged due to just the nature of the stomach. You then have inside your gastric glands cells called parietal cells. Now, parietal cells are going to produce intrinsic factor. They also produce hydrochloric acid. They also make a hormone called ghrelin. Now, ghrelin is an appetite-regulating hormone. When your stomach is empty, ghrelin production increases. And as a result, ghrelin is going to actually act on the brain and increase your sense of hunger. It's also going to increase the production of growth hormone releasing hormone by the hypothalamus in preparation for the arrival of nutrients. Because remember, growth hormone releasing hormone causes you to produce growth hormone from the anterior lobe of your pituitary. Growth hormone is going to cause your liver to then produce insulin-like growth factor. And growth hormone and insulin-like growth factor are going to basically help you grow. They're going to help you use your nutrients to increase your mass. Now, what's interesting is within 1 hour of eating, ghrelin secretion will actually stop. So after you have eaten, part of the reason why you don't feel hungry after you eat is because you don't have ghrelin being produced. In your gastric glands, you have chief cells. Chief cells are going to make things like pepsinogen, gastric lipase, and renin. You have mucous neck cells, which are going to produce mucus. And then you have what are called enteroendocrine cells. These cells are going to be producing hormones. And there are a variety of different types of enteroendocrine cells. We're just going to talk about two. We have what are called G cells and D cells. Now, your G cells are going to produce a hormone called gastrin in response to the presence of peptides inside your stomach. Now, gastrin, when it's produced, is going to help to stimulate the secretion of your gastric glands. So it's basically going to turn on your parietal and chief cells, which is something you want to do. If you have peptides in the stomach, it means that you have food in there. And if you have food in there, you need to start breaking it down. And the secretions of your parietal and chief cells are going to help you accomplish that. Gastrin also is going to help to stimulate contractions of the stomach wall. So it's going to help promote mechanical digestion within the stomach. It's also going to help mix your food content with all the secretions coming from the gastric glands. Gastrin then also travels to a structure a little bit away. It's at the junction of your small and large intestine. It's called your ileal orifice. And it's actually going to cause this orifice to open, and that's going to help facilitate the movement of residue, the leftovers, from your small intestine into your large intestine. Now, if gastrin sort of acts as this on switch for digestion in the stomach, the somatostatin produced by your D cells is going to act as an off switch. In fact, somatostatin can inhibit your G cells. So it inhibits gastrin secretion. It's going to suppress motility of the stomach. So it's going to slow the muscular contractions of the stomach. It's going to delay stomach emptying, meaning you're going to keep stomach contents within the stomach and you're going to stop moving the contents into your small intestine. It inhibits secretions by the pancreas. It inhibits gallbladder contraction and bile secretion. It also decreases blood circulation to your small intestine and decreases nutrient absorption within the small intestine as well. Now, let's look a little bit more specifically at the different secretions within your gastric juice. Now remember, salivary amylase was produced by the salivary glands and was part of your saliva, but your saliva gets swallowed with your bolus. So there is some salivary amylase in the stomach, and it will continue to do its job until you get to pH 4.5. And as the pH is dropping towards 4.5, this enzyme does start to work more slowly. So its action is declining, but once you hit 4.5, this enzyme is completely deactivated. The lingual lipase that was made by the tongue is finally going to be fully activated. It's going to start breaking down those triglycerides. The acidic environment of the stomach is due to the really nice industrial grade hydrochloric acid produced by your parietal cells. And this hydrochloric acid has a lot of different functions. It is providing non-specific defense against ingested pathogens. Many pathogens that are ingested cannot survive pH 2 to 3 acid, so very effective at protecting you. We use this acid to convert ferric ions into ferrous ions, which can then be absorbed and used by your body. If this does not sound familiar, please make sure you go back into chapter 18 on blood. We had a whole lecture section on the use and recycling of iron in the body. Your hydrochloric acid helps to denature your proteins. Now remember, denaturation from A&P1 Chapter 2 is the process that causes you to lose the three-dimensional structure of your protein. So basically, your nice big 3D proteins start to unravel. And once the proteins lose their shape, they lose their function. So we're going to use your hydrochloric acid to start denaturing the proteins that are in your food. Also inactivate any enzymes that happen to come in with your food. And once the proteins and enzymes are denatured and unraveled, they're in a state that can be digested. So this denaturation is necessary just so that you can break your proteins down chemically. We use the acid to break down your plant cell walls so that you can access the nutrients that are inside your plant cells, and to break down any connective tissues that you ate when you eat meat. So any cartilage, dense regular connective tissue, so some tendons, if you eat any ligaments, those connective tissues can all get broken down by the hydrochloric acid. The hydrochloric acid is going to be necessary for activating the lingual lipase, and it's also necessary to activate pepsinogen. Now, pepsinogen, as I mentioned on the previous slide, is produced by your chief cells. And pepsinogen is an inactive chemical. Okay, it is a protein, but it's inactive. It doesn't have function. However, when that pepsinogen enters into your gastric juice and interacts with that acidic environment, it is transformed into pepsin. And pepsin is an active enzyme. And the job of pepsin is to actually break down your proteins into smaller peptides. So pepsin is going to begin your process of protein digestion. Now, peptides are still too big to be absorbed, but at least we have begun breaking down those big proteins into something smaller. You have gastric lipase, and gastric lipase is very similar to lingual lipase in action. It's going to help break down your big triglycerides, which are your dietary fats, into fatty acids and monoglycerides. However, it is a really minor enzyme. In fact, with lingual lipase, these two enzymes only perform about 10 to 15% of your dietary fat digestion in an adult. However, gastric lipase is very significant in infants. In infants, it's responsible for about 50% of your lipid digestion. So, as an adult, very minor contribution, but in infants, very significant contribution. Renin is actually only found in infants. And what renin will do is it's going to curdle milk proteins. And by curdling the milk proteins in the stomach, it makes the milk, it takes it from being a liquid to a solid, and that solid form remains in your stomach longer, which is actually going to allow the infant's stomach enough time to break the proteins down and get all of those nutrients. You have to kind of remember that your infant systems are not functioning at peak capacity. Everything is going to take a little bit longer because your organs are just starting to work on their own. Um, Mom did most of this. She supplied the infant with all of the nutrients ready to be absorbed by the body. So now your digestive tract is finally trying to work on its own. Things take a little longer. But as an adult, there's no need for renin because as an adult, you can very effectively break down proteins within a reasonable period of time within the stomach. You have intrinsic factor, which I mentioned is necessary for vitamin B12 absorption. And what's going to happen is intrinsic factor is going to be put into your gastric juice. It will bind to the vitamin B12 that you ingested, forming a large complex that can then be absorbed within the small intestine. You also produce a whole lot of mucus, and this mucus is going to help to protect your stomach from enzyme and hydrochloric acid destruction.